Hi guys, welcome to my channel. So let's start off with what we need to know for exams. We need to know the definition of a gene and allele, the definition of a genotype and phenotype, the definition of principle of segregation, homozygote, heterozygote, dominance and recessiveness, and independent assessment. So we will start off with genetic crosses. For genetic crosses, we will firstly understand what monohybrid crosses are. Monohybrid crosses are crosses where we compare single characteristics. Okay, so let's have an example. Say we have a homozygous round seed that crosses with a homozygote ringle seed parent. Since it's homozygous, we will have the same allele, and since round seed is dominant, the allele will be capital letter R, and for the other parent, since it's wrinkled, the allele will be small letter r. So, for gametes, one allele of one parent will associate with two allele of the other parent. Then the other allele will also associate with two other allele of the other parent. Then we get the FY generation genotype. Since the genotype is capital letter R and small letter r for all the generation, the phenotype will be round seeds. So if we cross the F1 generation, one allele of one parent will associate with two allele of the other parent. Then the other allele also does the same. Then for F2 generation, these are the genotype that we get. And since we have three genotypes where we have a dominant letter R, the phenotype of the F1 generation will be 3 round and 1 wrinkle for the small letter RR. Okay, if the method that we use is confusing, we can use the Punnett square to get the result. So in this example, if we cross a homozygous round seed with a homozygote wrinkles seed, these are the genotypes that we get. So for the second concept, which is dihybrid crosses, this is where we compare two characteristics between parents. So for example, we cross a round and yellow seed parent with a wrinkled green seed parent. So for the genotype, round will be double letter R and yellow will be double letter Y. And if we cross the parents, this is the genotype that we get, and all the F1 generation will be round and yellow. So if we cross the F1 generation, these are the genotypes that we get using the Punnett square. Okay, these are the genotype that we get for crossing the true parents. Then for the phenotype of the F2 generation, we will have 9 round and yellow seeds, 3 round and green seeds, 3 wrinkled and green seeds and 1 wrinkle and green seed. Something to note, if we have a dominant letter R, regardless of the second allele, and also if we have a dominant letter Y, regardless of the second allele, we will have round and yellow phenotype. Again here, we will have round and green seed since we only have the small letter Y. And here we have only the small letter R and big letter Y. We will have wrinkled and yellow seed. And in this case, we only have small alleles. The phenotype will be wrinkled and green.